Okay. So we're going to do a review, so I'm not really going to ask you guys if you have any questions, because we can do that in a second. I just want to finish the last few slides of the chapter. So basically we've learned about DNA. You guys know that DNA is heritable units of information that's passed on from parents to offspring over generations. And so you probably watched, Jerry Springer, Maury Povich or whatever, and so you know that, when they're comparing two guys to see who the baby's daddy is, which one's going to be the daddy? The one with the closest DNA to the kid, or the one with the furthest away DNA from the kid? So you know that the more distantly related someone is to you, the more different their DNA should be, right? And so we can apply this not only to how we're all related as humans, but we can apply this to how we're related to other organisms as well. And so we call that process molecular evolution. And so we can use this for paternity and maternity testing, or we can use this for looking at origins for humans, or we can use this to looking at, origins for other species compared to humans, and so on and so forth. So it's pretty straightforward. The way that works is you just look at, either you can look at DNA, or you can look at amino acid sequences, right? There's 20 different letters. And so this example uses amino acid sequences and looks at the difference in hemoglobin. Remember, this is the beta chain, so how many types of hemoglobin are there in a functional hemoglobin molecule? You guys remember that? We talked about a quaternary structure. I'll take that as a no. So there's an alpha hemoglobin and a beta hemoglobin, and there's 4 subunits, 2 alphas and 2 betas, and they come together to make a functional hemoglobin. So this is the particular beta subunit, and you guys can go back and look at the quaternary structure of proteins in this same chapter, and you'll see that the human hemoglobin beta chain is 146 amino acids long, and if you look at every single human being that has a normal phenotype, so we're not talking about sickle cell, right, just normal, we all have the same hemoglobin. Why do you think that is? We don't have the same hair proteins, hair color proteins. Why not? Because if hemoglobin changed, it wouldn't carry oxygen anymore. What happens then if you get a mutation in hemoglobin and you can't carry oxygen? So you would expect that to be what we call highly conserved in a population, versus something that, what if you got a mutation in your hair gene and it caused you to have gray hair? You gonna die from that? So then that sort of gene is free to mutate without consequence. Does that make sense? So when you're looking at this, you have to look at stuff that's conserved, right? If you're looking at things that are far apart, or things that are variable, if you're looking at things that are not that are closely related, like within humans. If you were using hemoglobin to try to identify a criminal, would that work? Why not? Yeah, it would be like if everybody on Earth had brown hair and they said, what does the suspect look like? And you said, oh, they have brown hair. They'd be like, okay, yeah, that's great, thanks for the help. So we don't want to look at this, but we would look at something else that varies. And that's what Maury and Jerry and all those dudes look at when they're trying to find out who the baby daddy is. All right, so gorilla, if we look at gorilla, there's one variation in amino acid sequence. If we look at gibbons, which is another kind of monkey, there's 2. Rhesus monkeys, there's 8. And in fact, you guys know the blood group. We talk about the Rh positive and negative. So that stands for rhesus because it was first discovered in, can you guess? Rhesus monkeys. That's right. So when you talk about your blood type is O positive, that plus is the Rh factor, the rhesus factor. Mouse, 27, frog, 67. So based on this data, what do you think we're most closely related to? And what are we most distantly related to? There you go. It's not hard. And so you've probably seen evolutionary trees that have been developed off of this sort of data. But back in the old days, they used like differences in skull size or, you know, length of the tail or things like that. Today, generally people use DNA data because it's more reliable. The reason it's reliable, and we can use this to actually clock things, is remember we talked about radiation, and radiation has a certain decay half-life, right? And so in mutations, if you're within the same species, mutations generally occur at the same rate over time, just like the decay of DNA. So if you look at changes in DNA, you can estimate how far apart people are related to one another. So can we use that to figure out how old we are as a species? All of the different DNA on the planet of all humans. Right, so this was done. It was done by a graduate student. Her name was Rebecca Kahn. I actually knew her because we did the same kind of work in mitochondrial DNA when I was getting my master's degree. And she looked at all of the population of women and their mitochondria, and the reason she did that is because in humans, the egg is really big, right? And the sperm is small. And so the egg, all the mitochondria, remember all cells have to come from other cells. So all the mitochondria have to come from other mitochondria. So all the mitochondria that everyone here has came from the egg, which came from your mother. So all of your mitochondrial DNA came from your mother. So she looked at the differences between all the mitochondrial DNA from all the different populations on the planet. And she calculated not only how old we are as a species, but how far back we trace. Do you guys know what the answer is? What she estimated? Yeah, so that's about right. Somewhere between 150 and 200,000 years old. We're, as a species, we're actually a relatively young species. If we look at DNA between humans, our DNA difference is different by 0.1%, which is a pretty small amount. For every person, the biggest difference is 0.1%. If you look at all of the gorillas, their DNA is different by 4%. So they're much more older species than we are. And then, any idea where we originated from? Africa. That's exactly right. So there was another guy named Michael Nachman who did the same thing, except he did it with males, right? To see. So that paper was called Mitochondrial Eve, from the Bible story, and he was looking for what he called Y Adam, right? From the Bible story, and because all males in here, we inherited our Y chromosome from where? Our father, and so you can trace all of that back to, guess what he found out? How old are we as a population as far as on our male lineage? Good guess, 200,000 years. And where do we originate from? Africa. So scientists have done a bunch of experiments on this and found, based on DNA evidence, that there was a small population of humans. Maybe, estimates range from 100 to 10 humans that populated everyone on Earth. So it doesn't tell you what happened before then, because we could have existed before then and everybody died out, and then the Earth got repopulated just by those individuals in that small group. But we're all related. So we're all brothers and sisters. All right. Yeah, in fact, most of us have some percentage of Neanderthal DNA in us. So it depends on where you're from. Europeans have the most, about 4% Neanderthal DNA. And that means that there is some inbreeding between Homo sapiens and Neanderthals, even though they're considered different species, which doesn't fit the biological definition of a species. The species aren't supposed to be able to interbreed and produce viable offspring, like Napoleon Dynamite's favorite animal, the Liger.
I think I read that 20% of the Asian population is genetically related to Genghis Khan.
Yeah, so yeah, it's I don't think the number's that high. I think it's maybe 8 to 10%. Yeah, but it's pretty high. So Genghis Khan was a busy person, killing people and impregnating people. Yeah.
Well, it was long twice. He was in prison for 12 years and automatically he has 9 kids.
Yeah, I don't know that particular story.
That's the story.
Okay, well, I'll have to take your word for it. Okay, so that's all the material that we're going to cover for the exam. And so the rest of the time I've sort of budgeted to just do a review. So we can do this however you want. I can go back and go through the chapters individually and ask you questions or point out things you need to know, or I can ask you questions that I have, or you can ask me questions. It's up to you.
Can I throw a suggestion? Go with 4 and 5 detail and then
Okay,
do questions.
it's fine.
Are you okay with that?
Okay, so chapter 4 is relatively straightforward. There's really not a lot that you can do as far as like concepts. It's just basic memorization. So this chapter is on organic chemistry, so make sure that you guys know the names. Well, I mean, you should know by now that carbon forms 4 bonds, and its valence is 4. And the reason that all life is based on carbon is because you can make complex things out of it. And we talked about like the Tinker Toys. If you only had one hole on it, that'd be a lame set, because all you can do is put 2 wheels on a stick, and that's the end of your game. So I told you guys that you need to know the basic structures. So one carbon saturated with hydrogens is methane, 2 is ethane, 3 is propane, 4 is butane. And if I put a double bond in butane, it becomes butene, right? And then we number it by the location of the double bond, which honestly, I don't care that you know where the double bonds are in butene or whatever, but I do care that you guys know the number of the position of the carbons. I don't even care that you know the number of the positions of the carbons on the sugars, but what I do care about is the numbers of the positions on the sugar in DNA. So that's why I'm telling you the numbering scheme.
As 3 prime.
Yeah, you asked me to do chapter 5, so we'll hit that. Alright, but that's the term. So in methane, ethane, propane, butane. Right. And then if it has a double bond, you need to know it has an E and E in it, right? And then the numbering system of the bonds. Alright, so you should know the valence of the major ones. You know, I'm probably going to ask you, what are the 4 most common elements in living systems? And you should be able to answer that. What's going on? Okay, so how many bonds can hydrogen make? 1, carbon, oxygen, 2, nitrogen, 3, sometimes it gets weird, but generally 3. So that's the valence. We talked about carbon skeletons can vary in length, right? So we just talked about that, right? 1 is methane, 2 is ethane, 3 is propane. Right. Because the term valence, it refers to the number of unpaired electrons in its outer shell. So oxygen has 6, and electrons go individually, and then as they come around, they pair. So how many unpaired electrons are there? 2. Now, if I ask you the valence electrons, that's how many electrons are in its outer shell, and the answer for that is 6. So there's a big difference between the term valence and valence electrons. So you know that we can change the shape, right? We talked about so butane and isobutane has different shapes, but they're the same chemical structure. Are they different? If it has a different shape, it's going to have a different physical and chemical property. So these are different from one another, and then they can form ring structures too. And remember, we talked about the position of the bond. So this is 1, this is 2 butene. If I move this to here, what does that become? 3 butene? It's 1 because you can rotate it around. So then this chapter talks about isomers. So remember, iso means same, meros means part. So isomers are basically structures that are made of the same atoms, the parts. So there's 3 kinds: structural. This is a structural right here. Same parts, different shape. That's a structural. Then we have geometric. So geometric means that they have a double bond, and the reason that it's different is because that double bond can't rotate. So if it's on the same side, like this, what's it called? Cis. And if it's on different sides, trans. Good. And remember, we talked about the difference between alpha and beta glucose is this. That's why. And then in enantiomers, so it could be anything. It's like an X in algebra, right? Well, no, it doesn't have to be the same. Like it could be. We're talking about the complex structures in here, not hydrogens. So generally, it's going to be hydrogens like this, and then this could be, like a methyl group or something like that, and that would still be considered to be trans. On the test, it'll be obvious. I won't be tricky. It'll be straight up. It'll be something like this, right? But to answer your question, it doesn't have to. So I don't want you to call me a liar when you go and take organic chemistry and say, yeah, either that's not true, what Obermiller said. But for this class, I won't. I'm just teaching you guys enough chemistry so that we can talk about the other stuff, right? So that you understand what the difference is between these different glucoses. Because they look the same, right? If you didn't know the difference, you'd think tree bark and Doritos had the same nutritional content, which they don't, just so you know. So enantiomers, or the other term for this is optical isomer. So in medicine, they'll call this optical isomer. If you talk to a pharmacist or somebody that's, pharmacy rep or drug company, optical isomers are very common. Most of the drugs that are synthesized have left and right-handed forms, which they call S for sinister, the left-handed form, and then R for the right-handed form, which is rectus in Latin. In science, usually it's D for dexterous, which is right, and L, which is for left. I don't know why, but it just is. So that's one of those things where I'm still trying to figure out why they made the pH scale the way it was. Anyway, so remember the rule is that it's a central carbon with 4 different groups on it, right? If it only has 3, it doesn't have a left or right-handed form. So if it doesn't have a left or right-handed form, it's called achiral, not handed. And if it does, it's called chiral. That's just the Latin word for handed.
So how would you add something like that?
Single carbon is that have a left and a right-handed form? Is that an enantiomer? So in order to answer this, you have to know the functional groups, right? So what is that? What's that? So how many different groups are there? So then, does it have a left and right-handed form? What's the rule? Then the answer is no. This is not an enantiomer or an optical isomer. Is that? The answer is yes, because it has 1, 2, 3, 4 different groups. What is that, by the way? What's that called? Hydroxyl, right? So I might draw this out and ask you, what's that functional group? What's that functional group? What's that functional group? Just like we did in the lab. That's hydroxyl. Alcohol is a chemical that has a hydroxyl in the end of it. But this functional group is not called alcohol, it's called hydroxyl. You put alcohol on the test, that's wrong. So you know.
I just remembered I was reading the lecture.
So remember the rule, and I showed you the video, right? 3. This one only has 1, 2, 3 groups, so I can rotate that. This one has 4, and I can't. It's weird. I mean, it's almost impossible to visualize, and it's like one of those little puzzle tricks. You're like, no, there's no way that works out, but it really does chemically. And then the functional groups. So make sure you guys know these and you can recognize them on a molecule that I draw on the test. Right? So I'm not going to get tricky. I'm not going to be like, bury it somewhere in some molecule. It's going to be obvious. It's going to be just like in your lab. Remember that lab that had the R connected to an OH? What's that? Again, I'm not trying to be tricky, but it's important that you guys know these because you have to be able to predict what a drug's going to do if you give it to a patient. Otherwise,
So the difference between carbonyl and carboxyl is the carboxyl has the OH on it. Carbonyl doesn't.
right. So this is a carboxyl. And then this is a carbonyl group. Hang on one second. project students copy amplifying DNA, and I had to get them going. Okay, so this is a OH. It's a hydroxyl group. That's the name of it. It is an alcohol. The name of the compounds are alcohols, but that functional group is called a hydroxyl. Again, you're just going to have to memorize it, right? OH is hydroxyl. So these are carbonyls. Remember, if it's on the end, it's on the end. So this is a carbon. I drew this on a test. That's a carbonyl, no question about it. But what kind of carbonyl is it? That's an aldehyde. That's exactly right. What kind of carbonyl is that? That's a ketone, right? So you need to be able to pick that out. Carboxyl groups, they're easier because they're always the same. And they're always on the end. And we already talked about that. So that's the carboxylic acid. Why is it called an acid? These guys don't share well, right? Oxygen's selfish. It keeps the electron for itself. So the hydrogen falls off. What's the definition of an acid? Donates hydrogen H plus into a solution. Yeah, it was funny because I just watched an episode of Mythbusters where they were doing the Breaking Bad special. Did you guys see that? And they were trying to find out if you could take a body and pour. They used hydrofluoric acid in it, and if it would make the tub fall through. If you guys haven't seen it, you have no idea what I'm talking about. But anyway, they tried to reproduce it. So the whole half of the show was talking about the chemistry of acids, and I was like, too bad people didn't take my class before they watched this, because they would understand a lot better what's going on here. No. So anyway, that's why this is an acid, and generally things that have this are considered acids. Like vinegar is an acid, but it's not, you eat that, so it's not a strong acid. You wouldn't eat hydrochloric acid, I assume. Amino groups, they're NH2s, right? And they take hydrogens. Nitrogen can make extra bonds sometimes, and so it will pull bonds out. And in that case, so this one has now 1, 2, 3, 4. So that's considered ionized. And if you take hydrogens out of a solution, what are you considered? A base. So aminos are base groups. And then SHs are sulfhydryl groups. And what are those for? Stabilized proteins. That's exactly right. And then phosphate groups, those are part of DNA. Really important because it's part of the nucleotides, and we talked about that in chapter 5. So what's the charge? I just asked those students that are doing special projects, they copied DNA and now they're separating it out on a gel, so we have to put it to an electrical field. Which direction do you think the DNA goes? To the positive or to the negative? Right, because DNA is negatively charged, so which direction does it go? To the positive end, right? And this is how, they figure out who the baby's daddy is, or, who left their blood on the crime scene, because you're able to separate DNA out in this electrical field. And it's because of this phosphate group that you can do that. Alright, so that's it. Chapter 5. Yeah, that's going to be straight up stuff. I'm gonna ask you questions like, what's a chemical with an OH at the end of it, or whatever? What is that called? What's an NH2 called? I'm going to draw it on the test, and you'll need to be able to identify it. Yep. So I haven't finished the test yet, but it's looking to be somewhere between 50 and 60 questions. So roughly 10 questions per chapter, 10 to 12 questions per chapter. Last semester I gave them a 60 question multiple choice test, and most people finished it in under an hour. So the question, either you know it or you don't. You're not going to be able to stare at it and go, oh, I visualize the letter C, and it won't work that way. So either you know it or you don't. It's not going to be, for me it's easy. I know this is a lot of information, but this is nothing compared to anatomy. Wait till you guys have to learn all the parts of the body and all the muscles and tendons and the names of those in Latin and Greek. Okay, so this is chapter 5. We talked about that this whole chapter is based on large molecules, right? So we grouped them into 4 groups. The first ones are the carbohydrates, or the sugars, right? And then the second group was the lipids. The third group that we talked about was the proteins, and then the fourth group we talked about were the nucleic acids. So we'll go through so everyone understands that a polymer is, it's like building a necklace. You have the beads and you put them on a necklace. So the beads are the monomers, the building blocks, the parts that make up the necklace. Depending on what kind of parts you use, it could be a diamond necklace or a pearl necklace or tanzanite necklace or whatever. The parts are what dictates what kind of necklace you're building. So what's the reaction to put the parts together? Dehydration. And what's the reaction to break them apart? Hydrolysis. And it's all the same for all of these. There's only one of these that we talked about that there's no assembling or disassembling the parts. It's just changing the functional groups. Do you remember what that is? It is the lipids, the specific one. It's all made from cholesterol. Steroids, yeah. So those are the only ones that don't follow this rule, but all the other ones do. All right. So we talked about carbohydrates. Cholesterol is not, no, those are proteins. Yeah. So, yeah, and then that's another thing. You got all this stuff and it gets confusing, so you're just going to kind of have to sit down and sort it out in your head. And that just takes some time. Remember, if you mess up this test, you get another chance. So, don't freak out. I guess it's easier to say. So, monosaccharides, right? Carbohydrates are made out of sugars, and so the simple sugars or single sugars are monosaccharides, and then we have disaccharides. How many is that? 2, and then polysaccharides, many, right? And they're all different based on the parts that you use to build it. So we call the polysaccharides complex carbs, and generally the mono and the disaccharides are called simple carbs. Usually the structure is 1 to 2 to 1, so carbon, hydrogen, oxygen. All sugars end in what? OSE. Glucose is the most common. It's the one that we're going to talk about this whole semester. It's the one we're going to use for energy or whatever. It's the rawest, simplest, most direct form. It would be like if you had, racing fuel available to your car versus a barrel of crude oil. So everything else, you have to refine it, convert it, amp it up with octane. Glucose is the raw, pure form of energy. And so that's what we're going to focus on. It's a hexose. What does that mean? 6 what? Carbons, good. What if it had 5? What would we call it? What if it had 3? Good. Alright, so sugars almost always have a carbonyl group on them. So if it's on the end, what do we call it? Aldose. And if it's in the middle, so chances are I'm probably going to draw sugar. I'll tell you it's a sugar, and I'll be like, is this a ketose or an aldose? Is this a triose or a pentose? So as long as you can count the carbons and you know that if it's on the end versus in the middle, then you should be okay. Yeah, it'll be something like this, right? I'll put this up here. I won't have this or whatever, and I'll say, what is that? A triose, a pentose, an octose, a hexose. What is it? Pentose. And I'll ask you, is that an aldose or a ketose? And how do you know? It's on the end, right? What about this one? What is this? Describe this to me. Well, in the terms that we were just talking about. So it's a triose and a ketose. Good. All right. Remember the linkages. So on the test, I'm going to ask you stuff like, what kind of linkage is between that makes up amylose? Well, yeah, it's a glycosidic linkage, but remember, you need to know, is it alpha 1,4, 1,2, 1,6, beta? It's alpha 1,4. Good. What about, is it a straight chain? What about amylopectin? What are the bonds in it? Alpha 1,4s. Is it straight chain or branched? Branched. What about glycogen? It's alpha 1,4 and alpha 1,6. Is it straight or branched? Branched. Where's it found? Plants or animals? Animals, right? We store our energy as glycogen. Plants store their energy as amylose or starch or carbohydrates, yes. So that's the sort of stuff that you need to know. And then you can just go through the slides and look at that. Cellulose. What are the bonds in that? I switched it to glycogen so you couldn't cheat. Cellulose is beta 1,4. Straight or branched? Straight. Good. What about chitin? What's the bonds? Beta 1,4. Good. We didn't talk about if it's straight or branched, but it's straight. So that's what you guys need to know. Right? And I might ask you something like, you found an insect and you're curious about what its shell is made out of and what kind of bonds it's made out of. So you go to your biology teacher and ask them, what are they going to tell you? Yeah, beta 1,4. All right, so the lipids. Remember we talked about 3 classes: the triglycerides or triacylglycerols, which are the common fats like that are in your fat cells, right? That you store your energy as. And then we have the phospholipids that your cell membranes are made out of, and then we talked about the steroid lipids. So those are the 3 classes. So what are the fats made out of? What are the building blocks? What are the parts? Glycerol head, 3 fatty acid tails. What makes the fatty acid tails different? Well, that's true. But how do the fats differ? What's the difference between olive oil and canola oil? What's the difference between lard and peanut oil? Right, so it has to do with, so I might ask you, what sort of fatty acids would you expect to find in a brick of lard at the grocery store? Saturated or unsaturated? Saturated, that's very good. What about canola oil? Unsaturated, right? You wouldn't know mono or poly unless I told you. But what does it mean if it's mono unsaturated? It has 1 double bond in it. What about 2? So it's poly, right, unsaturated. Monounsaturated, polyunsaturated fats. remember, when we have a double bond, then we have that situation where we can have cis or trans. So I might draw out a fat and ask you if it's cis or trans. So make sure you know that. What's the function of fat? Why do we need fat? What's the purpose of it? Stores energy. What else? Protects your organs and yeah, insulation. Good. All right, phospholipids. They have a hydrophilic head and a hydrophobic tail. Why is this hydrophilic? Why can water surround it? Well, yeah, but it's not the bond. It's right. It's the charges. Remember, water can surround anything with a charge. Do they have charges? No, because they're carbons and hydrogens. The difference between those two is 0.4. So I don't care that you guys know all this. You don't even know that that it's called the phosphatidylcholine head. I don't care. What I want you to know is it has a head on it that is water loving and a tail that's water fearing, and that's what makes your membrane able to regulate, right, what's on the inside versus what's on the outside. What do we call that? Regulating a constant homeostasis. Good. So this is a meat cell. It's just the name. So this is what your cell looks like. It's a phospholipid bilayer. The tails go to the inside of the membrane, and the heads go to the outside where the water is. Right? Makes sense. So questions on the test might be like, if you looked at a cell, if you looked at, if you took a sample of tissue from an animal and you looked at it under the microscope, where would you expect to find the phospholipids? In the cell membrane, right? That's where they're located. What about the triglycerides? We didn't talk about that, but I just thought I might ask you. Well, no, they're stored in the cytoplasm. All right, and then we talked about steroids. All right, so those are all synthesized from. You can probably bet I'm going to ask you that because I'm a cholesterol buff. Right? So, what is it? What is testosterone synthesized from? What about estrogen? What about cortisol? Good. We didn't talk about cortisol, so I wouldn't ask you that. Okay, proteins. So we talked about what are the bonds between proteins called? Peptide bonds. And what are the building blocks for proteins? Amino acids, right? So this is an amino acid, right? This is a variable group. So the thing I was varying, right, I had an H up there and I put an O in front of it. So those are two different, actually, different amino acids. So this is an amino group. This is carboxylic acid, so it's called an amino acid. So I might draw that on the test and ask you, what is that? All you have to do is pick it up out of a lineup, right? So the peptide bonds, I might ask you what are the functions of proteins. I might ask you a couple questions like, do we use proteins for our immune system? All right. So what's the reaction to put amino acids together? Good. And then if we want to break them apart, hydrolysis. We talked about protein conformation, the 3D shape, right? So what are the 4 different levels of organization for shape for proteins? You guys remember? Primary, secondary, tertiary, quaternary. What's primary structure? is the linear sequence of amino acids. It's the order, yeah, that the amino acids come in. Just like the order of the letters in your textbook make the words mean something. So, what happens if I get a mutation in that? Will it change the primary structure? Yeah. What about secondary structure? So, secondary structure forms two categories. What are they? Alpha helix and beta sheet. What is it formed by? Hydrobonds where? In the R groups? In the backbone. Good. Tertiary structure. What is that? 3D shape caused by interactions in R groups. Excellent. Quaternary structure. Do all proteins have it? No. Only if you need two or more to put together to make it functional. So hemoglobin was an example that we used for that. So good. And remember, proteins, their shape is really, really important. Your body can tell the difference between a left and a right-handed molecule. That's pretty good. In primary structure, how do we number them? From the amino or the carboxyl end? The amino end. What's stronger, alpha helices or beta sheets? Why? Yeah, why? Yes, more hydrogen bonds, good. Alright, nucleic acids. So, this means DNA and RNA, essentially. Again, it's the order of the letters that are important. When I showed you guys the videos of the DNA to RNA to proteins. Two different kinds, DNA and RNA. So what's the difference between those two? Which one? Okay, which one has more or less oxygen? Ribose has one more oxygen. Deoxy has one less oxygen. Right? It's on the number 2 carbon. I'm not going to make you guys know that right now, but eventually you're going to have to know that because it has to do with how DNA gets copied, and it's really, really important. So there's the differences. Which one's found in DNA? What's sugar? Deoxyribose or ribose? Deoxyribose and RNA. Good. Where's the DNA found? The nucleus. DNA is turned into what? What kind of RNA? mRNA for messenger RNA. What's the other RNAs for? What's the tRNA for? Transfer RNA, what do they do? What's the purpose of it? It turns the messenger RNA code into the amino acid code, right? Their job is to transfer the message from mRNA to amino acids. What about rRNA? What's that? It's ribosomal RNA, and the ribosomes are what builds the proteins. They're the chefs, and so it needs to be able to read the message in the right order. So that's its job, is to make sure that is oriented correctly. So we talked about this. DNA is made into RNA, which made it into protein. Again, this is just memorization, so I might ask you, I'm not going to ask you what structure that is. I'd never draw that on a test and go, is that a T or U or C? I wouldn't ask you that. But I might ask you, like, which of these are purines? And then I'd put like AC, AG, TU, and, it's going to be 5 multiple choices, so if you can pick it out, then you'll get the answer right. And if you can't, get it wrong. So make sure you guys know that C's, T's, and U's are pyrimidines and A's and G's are purines. And just for the record, the difference is purines have 2 rings and pyrimidines have 1. I might ask you what makes up a nucleotide. What are the 3 parts of a nucleotide? Phosphate, sugar, right, and the nitrogenous base, or the base is fine. No, so that's the A, G, C, or T, right? It's these right here. And that's what makes them unique, because these are the same, right? If this is just a string of DNA, all of these green and yellow things are the same. There's no variation at all. So would that convey any information if it was all the same? No. So, I mean, that would be like having an alphabet with 1 letter in it. So what's the difference between DNA and RNA? I might ask you on a test. Which one's more stable? Which one is single-stranded? Which one has U's in it? Which one has T's in it? Which one has G's in it? Good. What does anti-parallel mean? They run opposite directions. How many bonds between A's and T's? Yeah. 2. How many between G's and C's? 3. Alright, it's right here. A's and T's, 2 hydrogen bonds. G's and C's, 3 hydrogen bonds. Yeah, RNA is single-stranded, so you would never have well, you could, but generally it doesn't happen. And then we just covered that evolution thing. So I might ask you on a test, we go to Mars and we find some species and we figure out that, it has blood and it has hemoglobin, and its hemoglobin is, this many differences between mouse and this many differences between parrot, and what is it more closely related to? Well, I didn't tell you how many mutations are in it, but that's the kind of question I'll ask you, right? How do we classify this organism we found on Mars? And obviously it's going to be most closely related to the one that has the least changes in, just like the baby daddy. Right? All right. You want some sample questions from those chapters? Uh-huh. Oh, yeah, we didn't talk about calculating the pH, so that's one of the questions that I need to get rid of, because we didn't cover that, so don't worry about it. But you will need to know stuff like, I'm telling you on the test, I'll put like, that's the concentration of H pluses. You did see quiz questions like this on the quiz probably. And so what's the pH of this? Yeah. So the quiz questions that you guys got, I'm in the process of trying to weed out some of the questions that come in the test bank and write my own and stuff like that. So this takes time, and I'm going to adjust the grades based on knowing that those questions are out there. So what I want you guys to focus on are the ones that we talked about in lecture. If we didn't cover it, I'm not a jerk, I'm not going to just throw stuff out there that we didn't cover. So I'm going to make you think about it. So you need to be able to use the knowledge that you know and figure out problems. But if we didn't talk about it, then it's not going to be on the test. So if I talked about it or put it on a PowerPoint, then it's fair game. So what is the pH of this? 1.0 times 10 to the negative 5th, which, because that's hydrogens, its pH Here's a good one. What is this? What's the answer?
So A and C and D.
So that, because it has the carbonyl on the end, is indeed an aldose, but it is also, because it has 6 carbons, it is also a hexose, so the correct answer is D. It's blinking, I can't, it won't turn back on. Sorry. Yeah. I'm not sure. So this question is, here's the general structure of amino acids. What functional group is this? And what functional group is that? So what's the answer? C. We didn't talk about that. That's a little more advanced. So which of these are not synthesized by dehydration reactions? Yeah, I just said it, steroids. Yep, that's right. So B. Which of these has branches of alpha 1,4 and alpha 1,6? Yeah, the answer is glycogen. Correct. So we talked a little bit about this. I mean, I didn't give you the straight up answer, but you should be able to deduce that. What's the difference between things you can digest and things you can't? What do you think? The answer is B, right? All the things we talked about have beta. I mean alpha, not beta. So you can't digest insect shells and you can't digest tree bark because it has beta linkages in it. Which of these is least diverse in its properties and structure? So which one varies the most? Which one varies the least? Which is the most boring molecule up here? Starch, because all it is glucose, 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 glucose. It's the same, right? Just over and over again. Polypeptides are amino acids, right? You can have all kinds of R groups in any order, right? Again, it's like having an alphabet with one letter. How would you read your book if it just said A? You know, versus varying the letters like DNA and RNA and proteins. All lipids are remember, all lipids aren't made of glycerol and fatty acids. Cholesterol is a lipid. That's not E. What they have in common is that they don't dissolve in water. Why? Because they're made of carbons and hydrogens. And those are what kind of bonds? Nonpolar. Yeah. Alright, we didn't talk about this specifically, but you should be able to deduce this based on what we talked about. So what do you think? What kind of oils are in tropical fish versus Arctic fish? So, so just, I wouldn't put this on a test, but just to give you a hint, your membranes have to, all of your fats have to stay at a certain fluidity. So, if you're colder, what would you want to make your fatty acids? Saturated makes it more solid, right? You want to make it more liquid at cooler temperatures. So that I wouldn't ask that. I might ask this, right? So remember we talked about what the difference is between DNA and proteins. So what molecule is found in, you want to label nucleic acids? What would you use? What kind of radioactive molecule? It's phosphates, right? Because all DNA has phosphate functional group backbones. What if I said proteins? What would it be? It's sulfur. Remember, I wouldn't ask this, but DNA has phosphates and proteins have sulfur. I will probably ask this later on when we start talking about DNA replication. Yeah, is that the only answer? Yes, the correct answer is all of those, because if you alter the primary and you don't alter the secondary, it wouldn't have an effect on the ultimate protein shape. So you know it has to alter the secondary. Stupid screens. Now we got to switch to this one. Okay, so this one is, how is RNA different from DNA? Sorry, you guys have to look over that one. Oh, it's still flashing. Yeah, the correct answer is D, right? It does all of that. That's how it's different from all of them. This, you may not know this word, right? But I mean, you know what a duplex is. Like a house, like a duplex. So it means it makes double strands. Alright. So we got 10 minutes. Pick what you want. Okay, what do you want to do?
How to figure it out? I know polarity is equal to moles per liters, right?
Yeah, moles of solute per liter of solvent? No. Solution. So, on the test I'm not going to make it very terribly hard. I might ask you. I want a one liter of a zero point 0.5 molar solution of sodium chloride. What do you do? So yeah, so the first thing you have to do is find the molecular weight of what your solute is, right? In this case, it's salt. But it could be, I could put glucose on there. Whatever it is, I'll probably give you the chemical formula for. So make sure you guys bring a periodic table with you if you can't see that, the numbers on it. I can't see this from here. I don't have my glasses on. So what's the molecular weight of this? It's the mass. 58.5, that's right. So sodium is what, 23? 22.99. Chlorine is 35.5. You add them together. Remember, that's Dalton's, but we just converted to grams. And this is a mole, remember? So we've just calculated a mole.
So is that means that is equal to 6.03.
If you shrunk yourself down, if I had a bowl with 58.5 grams of sodium chloride and I said, guess how many atoms are in this of salt? So we're talking about this together. How many of those are in my bowl? No, it's 6.022 times 10 to the 23rd, which is Avogadro's number. 6.0, well, 2, 2, 2, 3, 5, 6, 7, 1, 8, 3.141592653575. What?
Like there was a question where it was like,
Oh, yeah, that question is worded poorly. So the one that said, you have a liter and you pour 100 milliliters out, and then the answer is 6.022 times 10 to the 22nd, because you've taken 1/10. Like if I poured this out and I only gave you a tenth of it, then it wouldn't be 6.022 times 10 to the 23rd anymore. It would be, well, actually that would be to the negative 24th, because it'd be, no, 22nd, 24th, because it would be less. So 2 orders of magnitude difference. But the question didn't say, I read that question, and the reason that it was worded poorly is because it didn't say that you had a mole to begin with, right? It made some sort of weird assumption. It didn't tell you how much the original concentration was. So you would just have to make a bunch of assumptions. So I would never give you anything like that on the test. So
Freshly prepared 1 solution of glucose water. Carefully pour out 100 milliliters.
okay, wait. So you have a 1 mole solution.
Glucose in water.
Okay. So aqueous solution of glucose.
Correct.
then you pour down 100 milliliters. How many glucose molecules are included in that 100 milliliters?
All right. So here's the problem, right? I could have 20 milliliters of a 1 molar solution of glucose, or I could have 100 milliliters of a 1 molar solution of glucose. So if I poured it all in to 100 milliliter, the answer would be 6.022 times 10 to the 23rd, right? So the question didn't tell you how much you started with. It could have been 5 liters. It could have been, 15,000 liters, and that makes a big difference, because it's assuming that you have 1 liter, right? And because of that, now you've taken out a tenth, so you have a tenth less. So you just multiply it by, or divide it by 10, and that gives you your answer. But it's worded poorly, and so if I put that on the test, not that I would, but if I did, I would toss it because it's not worded correctly. There's no way to answer that without making the assumption that that's the case, and it never says that in the question. Yeah, so yeah, because I asked for 1 liter, then it's 0.5 moles, right, divided by 1. So if you cross all this out, it would be 0.5 divided by 1, which would be 0.5 moles. That's how you get the answer.
And then 15.5 divided by 2.
Right. So if I wanted 0.25, what would you divide it by? 4. Or you could multiply it by 0.25. It's the same thing. Because dividing that by 4 is the same as multiplying it by 1/4.
So that's more
Yeah, a liter. Now, if I said I want you to make 500 milliliters, right, or 0.5 liters of a 0.5 molar solution, how much would you need to do?
0.5 divided by 0.5.
Well, 0.5 times 0.5, which would be 0.25 times that. So it would be a fourth. So you just multiply the numbers in front. How much of a liter? How much of a molar? And that gives you your factor. So if I said I wanted 2 liters of a 0.5 molar solution, it would be 2 times 0.5, which would be 1. So it would be 58.5 grams. Right. If I said I wanted 0.2 liters of 0.5 molar solution, it would be 0.2 times 0.5, which would be a tenth, right? So 0.1 times that, or 5.85 grams. So no matter what I put in here, all you do is multiply those together. Remember, this has to be in liters, and a lot of times I could do this, and then you have to do the conversions like you did in the lab, because they have to be in the same units. This means per liter, not per milliliter. Any other questions? No? Why would it be? Oh, that's for my day class. Yeah, no. Yeah. So I had to cancel my Tuesday lab during the day because my kid was sick. So because they have the same lab on Thursday, I'm canceling them both, and I'm going to have them do a makeup lab. But you guys are split, so you didn't miss your Tuesday lab, right? I assume. So I'm going to be here on Thursday. So yes, you have a lab on Thursday. That was just for my Southern and Dawson class, not you guys. I better make sure I clarify that. All right, good luck. Don't forget, green Scantron, number 2 pencil, calculator, non-programmable, non-graphing, periodic table. No phone.
I'm taking my SIM card out.
No.
That was helpful going over chapter 5 like that. Because I read